In this video, we're going to look at the lipid bilayer. Now, amphiphilic molecules, which are molecules that have a polar and nonpolar region, will generally form micelles in water. The hydrophobic effect will push all the nonpolar parts together, and you have the hydrophilic parts interacting with water. The size and shape of these micelles can actually be predicted based on what the lipids look like. And it also depends on how many lipids you have. Too few lipids or too many lipids will result in water in the center cavity. Now, if your lipid happens to have two tails, like glycerophospholipids and sphingolipids do, you get the cell or bilayer, which is what we see in the cell membrane. Now, phospholipids can also form what's called a liposome. And these are large solvent-filled vesicles. And here we see a liposome. You see it is a double bilayer as well. And these are hundreds of angstroms in diameter, so bigger than proteins for the most part, or some proteins. And they're uniform in size, so you'll form the same size liposome every single time. And these are generally stable because you have the bilayer and you have your heads interacting with water, so there's no reason for them to be not stable. All the tails are interacting with each other. And this is how we really study biological membranes. This is our model of what a membrane looks like. And one way we think we can use liposomes is through drug delivery. It's hard to get drugs into cells, but if you have your drug in this liposome, the idea is this liposome will fuse into the membrane and then your drug will be delivered to your cells. Now, when we think about the phospholipid bilayer, you have to keep in mind that these are not motionless. They're very fluid. And the lipids in this bilayer have two different types of motion they can have. You can have a flip-flop motion, also called transverse, and lateral. So transverse is extremely slow because you need to have your polar head group go through this nonpolar medium, which doesn't happen very often. While lateral, which are just moving left to right, are extremely fast and happen all the time. You can move roughly 1 micrometer per second in a lipid bilayer. And you should think of the fluidity as light machine oil. That's what your lipid bilayer is like. And here we're seeing an image, a computer generated simulation, where you have your water molecules on the outside interacting with your head groups, your charged head groups, and on the inside you have your oil lipid tails all aggregating together. Now, the lipid bilayer has two phases it can be in, and this is temperature dependent. So you can have your more gel-like solid phase and your fluid phase. And the temperature at which you switch between these two phases, we call that the transition temperature. And this transition temperature is based on what type of lipid you have inside your membrane. So the longer the tail, and the higher the transition temperature. The more saturation, so the less double bonds, the higher the transition temperature, because it's harder to separate your molecules if they're long and if they're straight. And for the most membranes, this transition temperature happens between 10 and 40 degrees centigrade. Now, cholesterol actually increases this solid look. It decreases membrane fluidity because it will inhibit the ordering of the side chains, and it will push molecules together. So you could think of cholesterol as like wooden paddles that you put inside your membrane to shove your membranes together. That's it for fluidity in your cell membrane. I will see you in the next video.